So again, everybody, thank you for joining a presentation by Dr. Rachele Sangaletti, who is an assistant scientist in Department of Otolaryngology. She was a postdoc at Physiology and Biophysics for quite a number of years before joining my lab. And as many of you know, we're interested in studies of pathophysiology of the inner ear, so hearing and balance disorders. And she's been working in the lab looking at safety, efficacy, and mechanisms of neuroprotection afforded by mild therapeutic hypothermia in a rat model that we have developed for cochlear implant trauma. And really, like, we're hoping that you'll have lots and lots of good feedback for Rachele and me to take. And so I'll turn it over to Rachele.
Thank you very much, Dr. Rajguru, for the introduction. And thank you all for attending my first talk at the Miami project. I'm really excited to talk about my, my research today. So I would like to start showing you a cartoon of the auditory system, which is composed by 3 parts: an outer, a middle, and an inner ear part. And the talk today is going to focus on the inner ear, in particularly on the cochlear, which is shown here as a spiral cavity where we can find an important organ, which is called organ of Corti, which is important for the translation of the sound waves into electrical signals that are sent to the brain. So the functional properties of the cochlea are simplified if you think about the cochlea as a linear structure with 2 liquid-filled cavities divided by an elastic membrane, which is called basilar membrane. So the sounds vibration that gets to the middle ear are going to stimulate the cochlea, creating like a fluid motion that propagates along the 2 liquid-filled cavities. And this fluid motion creates like a wave propagation along the basilar membrane. And the beauty of the cochlea is that each portion of the basilar membrane gets excited upon different frequency stimulation. So for example, low frequency sounds are going to create a motion of the basilar membrane at the apex. Middle frequency sounds are going to excite the basilar membrane in its middle, and high frequency sounds are going to excite the basilar membrane at the basal part of the cochlea. So the motion of the basilar membrane stimulates the organ of Corti that consists of 3 layers of outer hair cells, 1 layer of inner hair cells, and 2 types of spiral ganglion neurons. So the vibration of the basilar membrane is going to stimulate the hair cells, bending a part of this cell that is called stereocilia that are located at the apex of these cells. And this electromechanical force is then translated into electrical impulses that are sent to the auditory nerve and up to the brain by the spiral ganglion neurons. So the loss of damage of any type of these cells causes the pathology of hearing loss. Hearing loss is a massive issue health worldwide, and there are different causes that brings to the hearing impairment. And for example, age-related hearing loss, infection, toxicity, genetic defects, noise, and physical trauma like the cochlear implantation trauma that is going to be the focus of my talk. And hearing loss is also a severe, has a severe social and economic consequences that we have to take in consideration. So the most effective treatment for hearing impairment is the cochlear implant device. The cochlear implant device was first implanted in the 60s, and since then the technology of these prostheses has tremendously increased. So what are the components and how the cochlear implant device works? There's a speech processor that picks up, and a microphone that picks up and digitizes the sounds that are then sent to a transmitter. The transmitter send the stimulus to a receiver that is implanted under the skin behind ear. And the receiver sends the electrical impulses to an electrode array that is surgically implanted inside of the cochlea. And the electrode sends the electrical stimuli to the auditory nerve, bypassing all the cells that are damaged in the cochlea. And this is on the right, is how a cochlear implant device looks like. So I'm sorry. So the first paper that talk about the electrical excitation of a cochlear nerve in man was published in 1957. And since then, the technology of the cochlear implant device, as I said before, has tremendously advanced. And as we can show here, from since the 80s to the 90s, there's been like an improvement of the understanding, of the speech understanding of the people that were implanted in with the cochlear implant device. And the same type of research was then performed later on patients that were implanted with the nucleus hybrid. The nucleus hybrid is a combination of hearing aid to amplify the low frequencies sounds with a cochlear implant device that allows the detection of high frequencies sounds. And here we can see that the patient that were unilaterally or bilaterally implanted have a progressive increase of understanding speech over a period of time of 5 years. So as for all the devices that have been surgically implanted, there are some risks associated with the cochlear implant insertion. And today I'm going to talk about the risk of a mechanical trauma. So the figure A is showing a part of the cochlear implant device, and in the red box is the electrode array that is surgically inserted inside of the cochlea by the surgeon. Figure B from 1 to 4 is the ideal insertion path of the electrode inside of the cochlea. So if the surgeon, which is blind during this procedure, pushes the electrode inside of the cochlea too quickly, especially in the first step, the electrode can hit the external lateral wall or the internal lateral wall of the cochlea. Non-traumatic electrode insertion can lead to a preservation of the residual hearing and the cochlear sensory neural cells that are located in the apex and in the middle of the cochlea, and cells that are responsible for the detection of the low frequencies and middle frequency sounds, respectively. And this is just a cartoon of a cross-section cochlea showing that the electrode is inserted in an opening made by the surgeon, which is called cochleostomy, inside of the cochlea through the scala tympani. The insertion happens at the base of the cochlea, and usually the electrode is pushed for 1.5 turn of the cochlea. Okay, so let's take a close look of the microscopic damages of an electrode insertion. This is a cartoon of our cochlear cross sections showing the different parts of the cochlea. So if the electrode insertion is too quick or is too rough, the electrode can hit the lateral wall, damaging the spiral ligament or the stria vascularis. Damages can happen to the spiral lamina or the basilar membrane where the organ of Corti is. Depending on the insertion depth and depending on the position of the electrode insertions, the mechanical trauma associated with the cochlear implant insertion can lead to acute phase of inflammatory responses, which involves the increase of vascular permeability, infiltration of macrophages and fibroblasts at the site of the injuries. Macrophages and fibroblasts releases inflammatory cytokines and chemokines that leads to an increase of reactive oxygen species, and apoptosis of the hair cells that are located at the middle or apex of the cochlea. Acute phase of the inflammatory response is usually followed by a chronic phase of inflammatory response, where the macrophages and fibroblasts releases growth factors that leads to a proliferation of fibroblasts with a formation of a fibrous scar tissues around the cochlear electrode. So there are several therapeutic interventions aimed to reduce the deleterious effect of cochlear implant trauma. And these strategies varies from several surgical approaches, different type of electrodes that are inserted inside of the cochlea, or pharmacological approaches like inhibitors of apoptosis, corticosteroids, or antioxidants. But none of these are 100% effective in reducing the damage caused by a cochlear electrode implantation. So in our research, we are taking in consideration of another therapeutic strategies, which is the mild therapeutic hypothermia. And as you know, mild therapeutic hypothermia is a worldwide treatment intervention shown to improve the outcomes of a variety of injuries, like spinal cord injury, cardiac arrest, traumatic brain injury, cerebral ischemia, noise-induced hearing loss, and cochlear implant-induced hearing loss. There are a few papers showing the efficacy of mild therapeutic hypothermia in preserving residual hearing post-cochlear implantation trauma. And the first paper was published in 2005 by Balkany and colleagues, where they used systemic hypothermia against the trauma caused by a cochlear implantation. Later on, our lab published a paper showing that localized therapeutic hypothermia conserves residual hearing in cochlear implantation. And in this research, we are using the device shown here in the figure A to deliver hypothermia at the cochlea. And the device is composed by a peristaltic pump that allows a continuous circulation of a saline solution through the system and through this hypothermia probe that is then placed next to the cochlea. The peltier part and the reservoir allows to keep the saline solution cold, and the thermoelectric controller allows us to set a specific temperature of the saline solution. The figure B showing the placement of the tip of the hypothermia probe next to the cochlea that is surgically exposed. The trauma is induced by the implantation of the electrode, insertion of the electrode inside of the round window, and we were able to measure the changing in the cochlea through a thermistor that was placed at the round window. The figure C showing the hypothermia protocol that was used during this study, which is 60 minutes of delivery of hypothermia delivered. And during these 6 minutes, the temperature of the saline solution is brought to from 30 Celsius degrees to 5 Celsius degrees. It's kept at 5 Celsius degrees for 20 minutes before and after the electrode insertion, and then the temperature is brought back to 30 Celsius degrees. In figure D, it's a graph showing the changes of round window temperature during these hypothermia protocols. Okay, so to test the efficacies of the localized mild therapeutic hypothermia, we performed auditory brainstem responses. And I'm gonna talk about a little bit about this non-invasive procedure where 5 electrodes are placed on the skin of the forehead and behind each ear. So the patient sits usually on an armchair in a quiet dim room. Headphones are given to the patient to deliver an array of acoustic stimuli that do not exceed 70 decibels. And the response that are recorded by the electrodes following the acoustic stimulation are then digitally stored. So, but what are these responses that the electrodes are recording? The responses are the acoustic, the auditory evoked potentials of neuronal activity along the auditory pathway. This is a cartoon of the auditory pathway that starts from the cochlea and goes up to the cortex, auditory cortex, for the conscious perception. Right here on the right, this is an example of an ABR, auditory brain response traces, and each wave corresponds to the neuronal activity that is being recorded following by acoustic stimulation in each part of the auditory pathway. For example, the wave 1 corresponds to the neuronal activity recorded from the distal cochlear nerve, wave 2 from the proximal cochlear nerve, and so on until we reach the auditory cortex. Okay, so from the ABRs, we can collect different type of information, and one of these is the hearing thresholds, which are the sounds level below which a person ear, or in our case, an animal model, is unable to detect any sound. Higher are the hearing thresholds, the more hearing loss the subject is experiencing. These graphs are the hearing thresholds recorded from the brown oval rats that were divided in 3 different groups. The control groups, which is the contralateral ears that is used as a control. The group that was where we implanted the cochlear electrode in normothermic condition, and a group, that received a cochlear implantation and mild therapeutic hypothermia. The hearing thresholds were recording at different different frequencies and at different time points before, which is the before the surgery, and up to 1 month post-surgery and hypothermia delivery. In our graph, we can see that if you follow the red line, so in normothermic cochlear implant groups, the hearing thresholds increase at day 1 post-surgery, and they remain elevated for the entire time of the study for all of the frequencies. Looking at the blue line, the hearing thresholds of the mild therapeutic hypothermia cochlear implant group were elevated at day 1 post-surgery, but they progressively decrease over time throughout all of the frequencies. So these data are showing that mild therapeutic hypothermia preserve residual hearing against cochlear implant trauma. After the functional study, the cochlear were collected for immunohistochemistry analysis, and from figure A to C shows the basilar membrane of control cochlea, B a cochlear where we induce a trauma in normothermic condition, and a basilar membrane of a cochlear where the trauma was induced and we delivered mild hypothermia. So as we can see in figure A, there is no damage at the cells of the sensory neural cells of the cochlea, whereas when we implanted the electrode, some of the cells started to be damaged or missing. With the cochlear, with the mild therapeutic treatment, we were able to preserve the hair cells. Figure D and figure E are showing the quantification of the outer and inner ear cells among the 3 experimental conditions. So the inner ear cells were not damaged following the cochlear implant trauma, whereas we can see a decrease of outer hair cells with cochlear implantation trauma, but mild therapeutic hypothermia was able to preserve these outer hair cells. And figure A is the quantification of the outer hair cells in the apical, middle, and basal part of the cochlea. There was no damage of the outer hair cells at the apical part, whereas in the middle and basal part, cochlear insertion causes the decrease of outer hair cells mild therapeutic hypothermia was able to preserve these other hair cells in the middle and basal part. So moving with the new results, I'm going to talk about first of the experimental designs. So we are using brown Norway rats, and these rats were divided in 4 groups. One group is the control group, unoperated. One group is the cochlear implanted in normothermic condition. One group received cochlear implant trauma and mild therapeutic hypothermia. And one group was treated only with mild therapeutic hypothermia, no cochlear implant trauma. We used the same hypothermia protocol, and we performed the functional study, auditory brainstem responses before and after, at different time points after the surgery and mild therapeutic hypothermia for a period of 3 months. We also performed RNA mechanistic studies. So we collected the cochlea 24 hours post-surgery and mild therapeutic hypothermia delivered. We extracted RNA and we performed a whole cochlear transcriptome analysis. Okay, so what we found. So here I'm showing hearing thresholds of comparing them between the 3 experimental groups shown here on top of the graph. And the hearing thresholds, as I mentioned before, were recorded at different frequencies and before surgery and up to 3 months post-surgery and hypothermia treatment. And as we observed before, in a normothermic cochlear implant group, the hearing threshold increased at day 1 post-surgery, and they remain elevated throughout the study. Whereas in a mild therapeutic hypothermia cochlear implant group, there's an increase of hearing thresholds at day 1 post-surgery, but the hearing threshold decreased over time throughout all of the frequencies. So these results are showing that, these data are showing that localized mild therapeutic hypothermia preserve residual hearing after cochlear implant trauma for a long period of time. We next wanted to establish, to see if mild therapeutic hypothermia was safe. And we compare here the hearing thresholds between the control group and the group that received hypothermia only. And what we can see is that the hearing thresholds of the group that received hypothermia only, so the blue line, is comparable to the hearing thresholds of the control group for all of the frequencies. So hypothermia is not effective, but it's also safe. Okay, so before talking about the mechanisms of protection afforded by mild therapeutic hypothermia in our model of cochlear implant trauma, I want to remind you what are the mechanisms of the cochlear implant trauma. So as I said before, the mechanical trauma induced by the electrode insertion is causing an acute phase of inflammatory response, where we have an increase of vascular permeability with infiltration of macrophages at the site of the injuries. Macrophages are releasing pro-inflammatory cytokines that and chemokines that leads to an increase of reactive oxygen species and leads to the death of the hair cells through apoptosis or necrosis. During this, after cochlear implant trauma, we can also have oxidative stress, like for example, changes in calcium homeostasis that leads to an increase of cytoplasmatic reactive oxygen species that leads to associated mechanisms of cell death. Okay, so we perform whole cochlear transcriptome analysis using 3 different experimental conditions, the control groups, normothermic cochlear implant group, and mild therapeutic cochlear implant group. And here what we can see are the principal component hierarchical clustering and heating map of all the genes for each replicates. And both of the figures are showing like a separation of between the 3 experimental conditions. Figure C are Venn diagrams of the unique and shared upregulated and downregulated genes among these comparison. So we compare normothermia cochlear implant versus control, the red group, mild therapeutic hypothermia cochlear implant versus normothermia cochlear implant, the blue group, and mild therapeutic hypothermia cochlear implant versus control, which is the yellow. So in the results that I'm going to show you right now, we started to look at the effects of the hypothermia on the two groups, the red one and the blue one. So let's start with the normothermia cochlear implant versus control group. So we use these differentially expressed upregulated and downregulated genes, and we perform gene ontology analysis. And here I'm showing you the most significant biological processes that we obtain from the upregulated, downregulated genes. And what we found is that cochlear implant induces the upregulation of genes that are associated with inflammatory and immuno responses, whereas the biological processes associated with downregulated genes were related to myelination, synaptic transmission, and neural circuit formation. We also took a look at the molecular function and cellular components and EEG pathways related to the upregulated, downregulated genes. So the molecular function, the most significant molecular function associated with the upregulated genes were are the chemokines activity, calcium-dependent protein bindings, ATPase bindings, just to name some of them. The cellular components are associated with sarcoplasmic reticulum, vesicle, cell periphery, and NADPDH oxidative complex and cells, whereas the EEG pathways are associated with inflammatory responses. What about the downregulated genes? So the molecular functions associated with the downregulated genes are the ATPase activity for the cellular components, synapses, and for the EEG pathways, dopaminergic, cholinergic, GABAergic, glutamaergic synapses. So next, we wanted to focus on the genes that were related specifically to these biological processes, and we grouped them in these categories shown here from B to F. So what we found is that cochlear implant induces the upregulation of pro-inflammatory and chemokines and cytokines. Induces the upregulation of genes that are associated with migration of macrophages, for example, or genes that are related to wound healing and secretion of extracellular matrix, or genes like TP73 that induce apoptosis, or cell that is endothelial cells activation. Cochlear implant trauma is causing the downregulation of genes that are involved in myelination, synaptic transmission, and neural circuit formation. I'm not going through all of them because they are like a good number of genes. So what are the mechanisms of the mild therapeutic hypothermia in our model of cochlear implant trauma? And before showing you our results, I want to just say that mild therapeutic hypothermia preserve the energy or protects the vascular capillarities. And it also dysregulates oxygen consumption, excessive toxicity, inflammatory cascade, cell death signaling. So there's a lot of pathways that are regulated by hypothermia. But what happened in our model of cochlear implant trauma? So we use these differentially expressed genes, 174, among the mild therapeutic hypothermia cochlear implant versus normothermia cochlear implant group, and we perform gene ontology analysis. Here I'm showing the most significant biological processes associated with these downregulated genes, And what we observe is that hypothermia decreases the genes associated with muscle contraction, thermosection, inflammatory and immune response, calcium homeostasis, and oxygen consumption. We also look at the molecular function, cellular component, and EEG pathways. And what we observe is that mild-therapeutic hypothermia is downregulates all the genes associated with chemokines activity, calcium ion binding, cytokines activity. Regarding the cellular components, we can see that the most significant ones are the sarcoplasmic reticulum, the cells, the vesicles. And for the EEG pathways, hypothermia affects the TNF-alpha signaling pathways, chemokine, calcium signaling pathway, and tight junction, just to name a few of them. And as we did before, we look at the genes that are associated with the biological processes, and we group them as followed here from figure B to figure G. So mild-therapeutic hypothermia is causes the downregulation of inflammatory chemokines, cytokines like CCL2, CCL7, just to name a few of them, affects the chemokine or cytokines that are involved in, sorry, it was a mistake. So these are the genes that are involved in the activation and migration of the macrophages. These are the genes, these are the pro-inflammatory cytokines and chemokines that are downregulated by mild-therapeutic hypothermia. Mild-therapeutic hypothermia affects also genes that are like associated with the blood-labyrinth barrier permeability, like DCN or MEUSTN1, for example, or genes that are associated with the secretion of extracellular matrix, or genes that here in panel D are associated with calcium ion homeostasis, genes that are associated with oxygen consumption that are oxidative stress related genes shown here in figure E. And hypothermia also induces the downregulation of genes shown here in figure F and G that are related to thermoception and response to muscle contraction. So the last things that we did was to compare all these, the expression level of all of these genes among the three comparison groups that we have. So let's look at the blue bars. These are the downregulated genes in mild-therapeutic hypothermia cochlear implant versus normothermia cochlear implant group. What happened to the expression level of these genes in the other two groups? So these genes are upregulated in normothermia cochlear implant versus control. So we are looking at the red bars, or they are not expressed. Whereas in the group of mild-therapeutic hypothermia cochlear implant versus control, only few of them are upregulated or they are not expressed. They are not expressed. So we can conclude from our results that localized mild-therapeutic hypothermia preserves residual hearing following cochlear insertion trauma. Localized mild-therapeutic hypothermia is safe and does not adversely affect inner ear function. And regarding the mechanistic part, mild-therapeutic hypothermia protects against cochlear implantation injury, downregulating multiple and possible independent pathways. And with that, I would like to thank my PI, Dr. Rajguru, my colleagues, all our collaborators. And thank you all for listening to my presentation. So, if there are questions.
Hello.
Hello.
Nice talk. I actually was paying attention. I wasn't reading my email. Could you put your slides back up?
Yes. 1 second.
And I wasn't clever enough to figure out the slide number, but maybe slide maybe 25. I'm going to guess you had a dendrogram comparing
Yeah.
It had a heat map comparing your
Yeah.
different individual experimental data.
Yeah, going back. Sorry.
There,
Here.
that's the slide.
Yes.
So you have this clustering here, and you have in the middle you have 3 animals that kind of clustered together. The Normothermia, one with the cold, and then you have another cluster with 4 animals. And so I'm sure you were hoping to have the Normothermia cluster together and the 4 animals that got the treatment cluster together, but that didn't happen. Do you have a hypothesis? Because it seems like there's 2 animals that almost look like an outlier or something. So what's going on there?
Yeah, so I think one is the variability of the animals or surgeries, for example, or my suspect is also during the RNA extraction, which I was learning, doing it, and probably I didn't do it quite well. And so I think that may cause what we can see here, the variability, all these this fact that the groups are not completely separate, so they don't cluster very well together. Yeah. And we can see from the heat map, there's a separation, but it's not quite like this distinct.
So do you know who Matt Danzy is?
Who?
Matt Danzy.
No.
He was a graduate student in our lab. Now he works in the Department of Genetics, and he's really awesome at looking at this kind of data. He may be able to help you come up with some, I know it's post hoc, but criteria for excluding some of these animals that clearly don't cluster correctly, and then your data might be less noisy and you might be able to get some more information out of it. I don't know.
Yeah. So I think, we already exclude, I think, one of the samples, and I think the way we did the analysis, so Dr. Lee's labs helped us through this phase, because it's not my expertise. And I think the way they did the analysis were, so they were considering based on the adjusted p-value and other parameters to avoid this, the inclusion of these genes that could cause like what is called noise. But yeah. But yeah, I can see your point.
Okay, so I guess my next question, now that you've done this analysis, and I understand you're using it trying both to understand what's going on and why the hypothermia is helpful, but also maybe looking for synergistic things that you could combine with that.
Correct.
What is your strategy for identifying synergistic treatments? How are you gonna look for that? I know that that's something that Jay and Nagi are very interested in doing.
Okay, so with this RNA-seq analysis, we needed we're getting like a big picture of the effect of the hypothermia. So now we have to move forward and try to focus on a specific pathways that are regulated by the hypothermia. So let's say, for example, apoptosis intrinsic pathways. We can maybe combine the hypothermia treatment with a specific pharmacological approach that is also inhibit these specific pathways. And based on the results, we can claim if it's an addictive or synergistic. So we will have to perform RT-PCRs, immunohistochemistry analysis, maybe other, probably RNA-seq analysis.
Okay, maybe I missed it. Something that we struggle with in our lab when we do these experiments, because they're so expensive, is we often have to guess when to do the sample. You know, how far after the injury, what's the best time to get the most information? How did you think through that to come up with the time points for when you were going to do your RNA-seq analysis?
Okay, so 24 hours, because looking at the functional studies, we can see that we have already a trauma. So the hearing thresholds were elevated. So we know that at 24 hours, there's a trauma. And at 24 hours, because it's the time frame where you have an acute, also inflammatory response. So where we're supposed to see the majority of the genes associated with the acute inflammatory response to be upregulated or activated. We are planning to look for different time frames, like 3 hours, 6 hours, 12 hours. And yeah, which is the range, well, 24, 48 hours is the range of the acute inflammatory response post electrode implantation.
I think Vance, really good questions. I think in terms of the sort of like the heat dendrograms that the question that you brought up, I think Rachele is right. What we're seeing is, like, with hypothermia is primarily sort of suppression of a lot of things, right? And so it seems sort of in between, really, normothermia versus like controls. We have another trauma model that Samantha in our lab has developed on noise-induced hearing loss, and there we see a lot more separation. And again, that might be because, Rachele has gotten a lot better at sort of extracting the RNA from the samples and being able to be sort of quick at that, et cetera. So that might have helped us in terms of that. And so we have a much better quality data in the other model that we have. In terms of the time point question, I think that's a good question too. And that's something that we struggle with all the time, right? What is 3 hours good or 6 hours good? Most of the time, we're calling up Dalton and Dalton, when are we going to see an effect, right? And then Dalton will be like, I don't know, maybe 6 hours is a good time to look. I don't know. So he's shaking his head too. But the reason why we sort of pick 24 hours is because between 24 to 48 hours is when you start to see hair cell death from apoptosis. And so if you're thinking that hair cells are going to be the primary sort of targets that you're looking at from preservation or trauma, then that was a good first guess to sort of start with, and then eventually look at other time points. And in terms of your last question about the combination therapy, right? I think that's a really good question too. And we were really hoping that we'll be able to use this specifically for that, right? Be able to understand some things. And we did learn some things that, it seems that, hypothermia does affect apoptotic pathways, but not particularly to the extent we would like caspase-1 driven pathway or something like that, right? And so we're looking at whether caspase inhibitors could be additive to hypothermia. And with John Barrett, we developed a cell model looking at sort of oxidative stress and combining hypothermia and caspase inhibitors sort of together. And what we found is that cell death was significantly reduced in that model, right? So we're hoping that maybe something like that might be potential synergistic therapy. But yeah, great points.
I think
Thank you.
Great questions. We struggled with combination therapy back in the 90s because specifically when we cooled a brain or spinal cord after injury, especially if we tried to delay the therapeutic window to hours after injury, we found that the hypothermia did not produce long-term protection. It only produced transient protection. So we spent a long time going, okay, let's give free radical scavengers on type of hypothermia. Let's give NMDA antagonists on top of hypothermia. Let's give calcium channel blockers on top of hypothermia. And there's the literature there with hypothermia showing that we could actually extend the therapeutic window and produce long-term protection. So it's a really good area. I mean, it's encouraging the positive results you're seeing with hypothermia alone in this particular injury model. But again, you're cooling very early. So will you be able to do that in the operating room? I guess you will. It would be a unique situation where you can start cooling very early versus having to delay after a patient comes into the emergency room. But congratulations on the work that you've done. It just reemphasizes that hypothermia is kind of non-specific and affects many, many, many pathophysiological mechanisms in terms of neuronal, glial, vascular, inflammatory. And sometimes we get criticized in grants because, you know, how can you do science on hypothermia when it affects so many different things? And so it's interesting. We've put out most of our focus recently on inflammatory cascades because they seem to be very, very sensitive. But it's great to see this progress using temperature modification. And I've talked about this. The other aspect of this is many patients, you know, even the inner ear, they may have mild elevations in temperature. And so you would do a different experiment where you're actually doing delayed mild hyperthermia and seeing that it aggravates these things. So that's something else to consider for the future. But it's certainly a very, very nice strategy going forward.
Thank you, Dr. Dietrich.
Yeah, no, I think you're right, Dalton. There is some literature, only a few papers, but that show that in hyperthermic mice, when you expose them to louder noise, they tend to have worse outcomes, right? So temperature certainly has a lot of effects. And like, you know, in the auditory system, we have known that temperature is very critical, even in terms of driving neuronal responses and stuff like that. So, so I think it's certainly an area to be considered. But you're right. Like, we also get criticized a lot that, you know, it's just so, it's affecting everything. So how can you do? What's your scientific hypothesis, right? Like, you know, what's the specific pathway? Like, tell me about one pathway, and that's challenging.
Do you have a model for why it affects the type 2 hair cells, but not the type 1 hair cells? It's kind of curious that most of the damage was to the type 2s.
The outer hair cells?
Yeah, the outer hair cells, I'm sorry, yeah.
No, a model of, you mean the mechanistic model?
Or just, yeah, why the outer ones are affected without affecting the inner hair cells.
Yeah, it's been shown that are the first cells that are like get damaged or that die through the process. The inner cells, that is what follows. No, I don't have right now a mechanistic explanation of it. It would be nice to do like a single cell RNA-seq in the future. That could give us like more information regarding of the death of these hair cells. Yeah, but right now, no, because it's the RNA-seq analysis that we was performed on the whole cochlea. So we didn't do a distinction of hair cells, paraganglion neurons, or other structures.
Yeah, and maybe just the outer ones are more sensitive to damage from noise also, I guess. Maybe they're just more sensitive cells. I don't know.
Yes, because we have to consider that during the cochlear implant surgery, there's the trauma induced by the electrode, but there's also trauma that could be induced during the surgery by drilling the bones, the bullas, which is the bone that surrounds the cochlear, or if the electrode insertion is not done through the round window bodies, if it's done through the cochleostomy, then we have to make that specific opening, and it's another drilling part. So there's a different type of trauma that can be associated with the hearing loss, not only cochlear implant trauma, depending on how the surgeon is, if the surgery is soft or the surgeon, it's more rough during the surgery.
Now you might be able to distinguish damage from vibration versus damage from direct physically hitting the cells by looking at different places along the basilar membrane.
Yeah,
The one spot in the apex wouldn't be hit probably.
yeah, exactly. For example, if you perform the cochleostomy, which is at the base of the cochlea, is where you can damage residual hearing or cells at the base of the cochlea, whereas the cochlear implant trauma extends the trauma, the deleterious effect, the middle and apex part of the cochlea, and is where we're trying to focus on.
Wow, wow.
Yeah, I mean, like, the patients likely have low frequency hearing, right? So they likely have, the patients that we are targeting are the ones that have apical hair cells, but not in the middle and the basal turn. And surprisingly, what's been observed is that even if you put the cochlear implant mostly in the basal turn, hair cells on the apex also get inflammatory effects, right? And they end up sort of dying. So there are mechanisms that are sort of that really need to be identified, like, what it is. And that's partly why I think hypothermia is as effective, because it really sort of cools the whole cochlea rather than the drug that is only injected in the basal region or a middle region and doesn't really make it all the way to the apex.
Other questions, suggestions, concerns?
Yeah, very tricky to find which genes, as you were pointing out, are really causing the beneficial effect, or possibly something that is not a change in gene expression is operating to cause the beneficial effect at a very early time, because probably no genes would be upregulated very much for a couple hours, and it's possible that some of the major damaging processes are going on during those first 2 hours, although I guess the inflammation would be later. So
It's right after the trauma, up to 48, between 24, 48 hours.
So I'm not an expert in this, but this is kind of stuff has been studied intensely in the growth factor field and has relations to cell death. And immediate early genes get turned on in minutes.
Wow. Okay.
Yes.
Yeah, it'd be interesting to look at some of those that would closely. It's just, I guess it's tricky because the inflammation, the damage of the inflammation can be occurring much later, and the gene changes that are initiating that, maybe from intermediate early genes, might be starting very soon during the time of the hypothermia.
Yeah.
Or it could be a other damaging process during hypothermia that's then leading to the initiation of the inflammatory response. Anyway, it's really fantastic that you get such a great effect from the hypothermia.
Yes. Thank you.
Yeah.
Good job, Rachele. Thank you for a nice
Yes, great talk.
Thank you all for attending. I think we can stop recording, I guess, if there are no questions.
Yep.
Which